Bismillahirrahmanirrahim. Assalamu alaikum. I am Professor Dr. Mahmouda Begum, Professor and Head, Department of Anatomy, Adina Women's Medical College, Mokbajar, Dhaka. Dear students, you know now the world is passing crucial moment. We are all staying at home to save our country and people. I hope Allah subhanahu wa ta'ala give us a good morning and rahmat. Alhamdulillah, we are trying to continue our online teaching that should be beneficial for you. According to this activity, I would like to start my next session, second week of development. I had already completed the first week of development. Second week of development, part of germinal period. Because germinal period extend from first week to third week of development. Today, I like to let you know the day 8 and 9 of second week of development. Second week of development. Actually, the week, the main focus to form bilaminar germ discs. That is the yellow color and blue color. This is two germ layer. That is the two layer. This is known as the bilaminar germ discs. Before we start this week, we memorize this is the blastocyst and this is the embryonic pole, embryoblast, and surrounding the trophoblast, blastocele cavity. And this is the embryonic pole which erode the and gradually embedding the endometrium of uterus. So this is the uterine cavity, the blastocele gradually embedding the functional layer of endometrium. A day by day, there is a different major events occur in the second week of development. We know the embryoblast, inner cell mass. This is the embryoblast, blue color surrounding. This is the trophoblast, flattened epithelial cell. So day 8, second week start. This is day 8. Day 8, the embryo, that is blastocyst, partially embedding the endometrial stroma, endometrium of the uterus. In the stroma of the endometrium of the uterus, partially it is embedding. First event, the partially embedding. First week a touch and adhesion, and the second week start, that is day 8, there is a partially embedding the blastocyst, in the endometrium. Now the first event and most important, the area of the over the embryoblast. What the embryoblast, the trophoblast differentiate into two layers. So first is the site. Site is the over the embryoblast. The trophoblast differentiate into two layers. Inner layer, mononucleated inner layer. This is the inner layer, mononucleated cytotrophoblast, and the outer multinucleated without distinct cell membrane syncytotrophoblast. This is the multiple nucleated. This cell have the single cell has no definite boundary. So these are the syncytotrophoblast. So trophoblast differentiate into two layers: inner mononucleated distinct boundary of the cell, cytotrophoblast, and outer multinucleated node without distinct outer membrane. This is the cytotrophoblast. The trophoblast differentiate into two layer: inner mononucleated cytotrophoblast and outer multinucleated without distinct cell membrane, known as the syncytotrophoblast. The first event, that is embedding is the partially, it is the process of the implantation gradually occur to complete, but event the embryoblast and trophoblast. So trophoblast first, the overlap the embryoblast, those regions trophoblast is first differentiated into two inner mononucleated distinct boundary of the cell presence, cytotrophoblast, and outer multinucleated without distinct cell membrane, known as the syncytotrophoblast. So again memorize cytotrophoblast and syncytotrophoblast. Cytotrophoblast mononucleated and syncytotrophoblast multinucleated. Most important information, the mitotic figures are found in the cytotrophoblast but not in the syncytotrophoblast. So where the cell division is occur, which layer of the trophoblast is responsible for the mitotic cell divisions? Cytotrophoblast. And syncytotrophoblast there is no cell divisions occur. So cells of the cytotrophoblast divide. These cells migrate to the syncytotrophoblast, and where they are fused, one cell each other fuse and lost their individual cell membrane and form a syncytium, and known as the syncytotrophoblast. So mitotic cell division present in the cytotrophoblast, not in the cell of the cytotrophoblast migrate to the syncytotrophoblast layer, where they are fused and form multinucleated, no definitive cell membrane, form a syncytium known as the syncytotrophoblast. Now the cell of the inner cell mass, that is embryoblast, this is the inner cell mass. These are the inner cell mass, this embryoblast. These embryoblasts also differentiate into two layers. First is the small cuboidal yellow color. Small cuboidal cell, this is the yellow color. Hypoblast, a small cuboidal cell. This is adjacent to this is the blastocele cavity, known as the hypoblast. And others is the long columnar, that is a blue color. This is a long columnar cell. This is the epiblast, higher columnar cell, epiblast. And two layer together, the layers form a flat disc, bilaminar disc. This is the bilaminar disc. So embryoblast also divided. One is the small cuboidal cell close to the blastocele, it is the hypoblast, and another is the long columnar cell, it is the epiblast. So embryoblast divided but differentiate into two layers. This is the hypoblast and this is the hypoblast, blastocele hypoblast, and this is the epiblast, two layer. So trophoblast also two layer, embryoblast also two layer. Trophoblast divided into cytotrophoblast and cytotrophoblast, and embryoblast divided into hypoblast and epiblast. And there is one cavity appeared within the epiblast. This is known as the amniotic cavity. So blastocyst cavity, and this is the amniotic cavity. Amniotic cavity appear within the epiblast, blastocele close to the hypoblast. So hypoblast adjacent to the blastocele, epiblast adjacent to amniotic cavity. So the cell, epiblast cell, this is the epiblast cell adjacent to the cytotrophoblast. These are the epiblast and adjacent to the cytotrophoblast. This cell is known as the amnioblast. So the other cell, other epiblast, this is surrounding the other epiblast. These are surrounding the amniotic cavity. Amniotic cavity. So day 8 is another event. Amniotic cavity is formed within the epiblast, and cell adjacent to the cytotrophoblast is known as the amnioblast. So if there is an asking question, amniotic cavity is lined by epiblast, blastocele lined by, there is a blastocele, close to the blastocele there is a cell is the hypoblast. This is the hypoblast and this is the epiblast. This is the amniotic cavity. Now, in these conditions, when the endometrium in the day 8, the endometrium is more edematous, the site of the implantations, and highly vascular. And the gland, this is the endometrial gland. This is the endometrial gland. This is the endometrial gland. The tortuous, large, the secrete abundant glycogen and mucus. These are the uterine gland. They secrete the abundant glycogen and mucus for nutrition of the embryo. Day 8. So if we summarize the day 8, what are the events occurred in the day 8? First is the blastocyst partially embedding. First is the day 8, not complete embedding is not complete, partial embedding. Trophoblast differentiate 2 layer, cytotrophoblast, syncytotrophoblast. Embryoblast differentiate into 2 layer, hypoblast and epiblast. And small cavity appears within the embryoblast known as the amniotic cavity. Now day 9. During this day, the blastocyst is more embedded, deeply embedded, the blastocyst, deeply embedded endometrium of uterus, and the penetration site gradually closed by fibrin plaque, fibrous tissue. So day 9, the blastocyst deeply embedded in the endometrium of uterus. And also the trophoblast considerably progress in development. During day 9, trophoblast progressively in development, particularly in the embryonic pole. This is the embryonic pole. This region, the trophoblast, shows considerable progress in development. There also appear small vacuole. This is the small vacuole This is the small vacuole appear in the syncytium. Which layer? In the syncytium. And these vacuoles fused with each other and form large lacuna. So this phase is ultimately called lacunar stage. So second event, that in the syncytium or syncytotrophoblast at the embryonic pole, they are present gradually, there occur small vacuole. These vacuole gradually fused with each other and form large lacuna. So this stage is known as the lacunar stage. The first, blastocyst deeply embedding in the endometrium. Second, lacuna appear. This is lacunar stage. Another events in the day 9 at abembryonic pole. This is the abembryonic pole. Small flat cell appear. This cell originate from hypoblast and form a membrane along the blastocele cavity. This membrane is known as the exocoelomic or Husserl's membrane. So these are the Husserl's membrane. This membrane lined by flat epithelial cell. This cell arises from hypoblast, and this cell also line the inner surface of the cytotrophoblast, inner side of the cytotrophoblast. So the membrane, this membrane, exocoelomic membrane, and there is a hypoblast, is closed the cavity. This cavity is known as the exocoelomic cavity or primitive yolk sac. So this is the primitive yolk sac and this is the amniotic cavities. Two cavity. There is also two cavity. One is the amniotic cavity, another is the exocoelomic or primitive yolk sac. So first there is a deeply embedding and penetration site is closed by fibrin plaque. Then embryonic pole, in the embryonic pole trophoblast, there are present lacuna, and abembryonic plane, embryonic pole, there is a form another membrane known as the Husserl's membrane or exocoelomic membrane. This membrane and hypoblast surrounded the cavity. This cavity now known as the exocoelomic cavity or primitive cavity. This figure, day of 9. There is first is a leveling is the lacuna. This is the lacuna, trophoblastic lacuna. This is the trophoblastic lacuna, and this is the amniotic cavity, epiblast, hypoblast, exocoelomic or primitive yolk sac, Husserl's membrane, and this is the cytotrophoblast, and this is syncytotrophoblast. This is the cytotrophoblast, and penetration site is covered by fibrin plaque. Now summarize the day 9. Blastocyst deeply embedded, penetration defect closed by fibrin plaque, vacuoles appear in the syncytium, and vacuole closed fused with each other form lacunar stage, and exocoelomic or Husserl's membrane appear, formation of the exocoelomic cavity or primitive yolk sac. This is end of my class. Dear students, you are requested to share your any problem or inquiry of my online lecture class with your respected best teacher of anatomy department. Thank you all.